Hello, welcome to Biology, the scientific study of life. Phylogenetic trees as a hypothesis. This is a good point at which to reiterate that any phylogenetic tree represents a hypothesis about how the various organisms in the tree are related to one another. The best hypothesis is one that best fits all the available data. A phylogenetic hypothesis may be modified when new evidence compels systematists to revise their trees. Indeed, many older phylogenetic hypotheses have been changed or rejected since the introduction of molecular methods for comparing species and tracing phylogenes. Notice that research method applying parsimony to a problem in molecular systematics. Notice that. Application in considering possible phylogenes for a group of species. Systematists compare molecular data for the species. The most efficient way to study the various phylogenetic hypotheses is to begin by first considering the most parsimonious, that is, which hypothesis requires the fewest total evolutionary events, molecular changes, to have occurred. Technique. First, follow the numbered steps as we apply the principle of parsimony to a hypothetical phylogenetic problem involving four closely related bird species. First, draw the possible phylogenetic for the species. Only 3 of the 15 possibilities relating these 4 species are shown here. Notice that. To tabulate the molecular data for the species in this simplified example. The data represents a DNA sequences. Notice that consisting of just 7 nucleotide bases. 3. Now focus on site 1 in the DNA sequence. A single base change event marked by the crossbar in the branch leading to species 1 is sufficient to account for the site 1 data. Notice that. For continuing the comparison of bases at site 2, 3, and 4 reveals. Notice that each of these possibilities requires a total of 4 base change events marked again by crossbars. Thus, the first 4 sites in this DNA sequence do not help us identify the most parsimonious tree. After analyzing sites 5 and 6, we find that the first tree requires fewer evolutionary events than the other 2 trees. 2 base changes variances. For note that in these diagrams we assume that the common ancestor had GG. Notice that at size 5 and 6, but even if we started with an AA ancestor, the first tree still would require only 2 changes, while 4 changes would be required to make the other hypothesis work. Keep in mind that parsimony only considers the total number of events, not the particular nature of events, how likely the particular base changes are to occur. At site 7, the 3 trees also differ in the number of evolutionary events required to explain the DNA data results. Notice that results to identify the most parsimonious tree, we total all the base change events noted in steps 3 to 6. Notice that We conclude that the first tree is the most parsimonious of these three possible phylogenes, but now we must complete our search by investigating the 12 other possible trees. Often, in the absence of conflicting information, the most parsimonious tree is also the most likely, but sometimes there is compelling evidence that the best hypothesis is not the most parsimonious. Nature does not always take the simplest course. Perhaps the particular morphological or molecular character we are using to sort taxa actually evolve multiple times. For example, both birds and mammals have hearts with 4 chambers, whereas lizards, snakes, and turtles have hearts with 3 chambers. The parsimonious assumption would be that the 4-chambered heart evolved once and was present in an ancestor common to birds and mammals, but not to lizards, snakes, and turtles. However, abundant evidence indicates that birds are more closely related to lizards, snakes, and turtles than they are to mammals. Thus, the 4-chambered heart appears to have evolved independently in birds and mammals. Indeed, studies have shown that the 4-chambered hearts of birds and mammals develop differently, which supports the hypothesis that they evolved independently. The apparently parsimonious tree in this figure, notice that A and B, is not consistent with these facts, while the tree in the figure in this figure or the next figure, in this figure B, lizard, bird clades, notice that it's consistent with the additional facts. In this example, the problem is not so much with the principle of parsimony as it is with the analogy-homology issue. The 4-chambered hearts of birds and mammals turn out to be analogous, not homologous. Matching changes of bases in DNA sequences in 2 species can also occur independently, but the more bases that are involved, the less likely is that it is that the matching changes are coincidental. Applying parsimony in molecular systematics is more reliable for a data set of many long DNA sequences than for a smaller data set. Similarly, occasionally misjudging an analogous similarity in morphology as a shared derived homologous character is less likely to distort a phylogenetic tree if each clade in the tree is defined by several derived characters. The strongest phylogenetic hypotheses are those supported by multiple lines of molecular and morphological evidence, as well as by fossil evidence.